Hello, here's just a short lecture about the neural basis of dynamical field theory, the neural foundations of dynamical field theory. A lot of it was covered partially in previous classes, so this is just a little concentrated form of talking about that. Now, you know that in DFT, peaks are the units of representation, so we're thinking about activation fields that are defined over some spaces, some metric spaces that could be viewed as spanning the contents of the representations, so space, movement parameters, feature dimensions, and so on. We looked at different examples. While activation is the representation, it is the information about that contents or the probability of it, or if it's a perceptual decision, for instance, or the certainty we have about it or something. These could be functional significances or functional realizations of what activation amounts to, compared to not having a peak. So when you have a peak, it's always two things. It's that the value is specified along the dimension and that there is activation versus no activation, right? That means you have a detection. So that's the conceptual thing that we used. And so how does that link to neurophysiology? Now, that ultimately has to do with what does actually mean to have a neural representation. That word is used very loosely. And as I've argued a couple of times, it's always about how an inner state in the nervous system activation, population activation, relates to some state outside the nervous system, some sensory stimulus or some motor state, for instance, or environmental state in the world. And the notion that dynamical field theory exploits to talk about that relationship is that of tuning, of tuning curves. So a tuning curve is a function that links something outside the nervous system, here the feature dimension of some stimulus, to something inside the nervous system, the spike rate, for instance, of a particular neuron. And I illustrated in the course in which I introduced neural fields that the forward connectivity from the sensory surface to a neuron can give rise to such a dependence to the variation of firing as a function as you change the stimulus. And similar for the motor system, that the connectivity from a brain to a motor system could lead to such a dependence between the spike rate and some like movement parameter. Here is this historical example that I showed in some discussions we had from the original work that really was sort of trailblazing for a lot of thinking about higher nervous function, about the brain. What Huben Wiesel did is record from the primary visual cortex in, this is actually from monkey, where they were shown moving light bars onto the retina of the monkey. Monkey was just fixating this, actually anesthetized animal. And particular regions of the retina were sensitive, or when the line was shone onto particular regions, sketched here by this rectangle with the dashed lines, only light that fell into this part of the retina would actually induce any spiking, shown here by these marks, these vertical potentials recorded from an electrode inside the tissue, would elicit activity in a particular neuron in the visual cortex. And so this part of the retinal array is called the receptive field, the field where that neuron is receptive to visual stimulation. And moreover, the firing of that neuron was modulated with certain parameters of the visual stimulus. For instance, when this visual stimulus was a longish slit of light that was moved along its orthogonal to its long axis, then there were particular orientations of that line combined here with movement direction of that line, where the firing was vivid, while other orientations was no firing. So if you systematically vary that, this is the variation of that orientation angle that covaries here with movement direction and the firing of that neuron. You see that there's a very nice, smooth tuning curve. Let's call it tuning curve that shows how much the firing rate of that neuron is modulated with that parameter, that feature parameter, that local orientation, local movement direction parameter. And you can do these kind of tuning curves for many different aspects of visual display. And this is the analogous procedure in the motor domain. This goes back to, comes back from the Jacopoulos lab. So here a monkey makes a movement, and more like you have to make, train an animal to make a movement. And this is while there's recording in the motor cortex, primary motor cortex. And the monkey actually had learned to move to one of 8 targets. It's a horizontal movement of the hand, some arrangement. The monkey has learned to do that movement at a particular time that is indicated by a cue. And then while the monkey is moving, experimenters have electrodes in the motor cortex and try to find neurons that vary their firing while the monkey is preparing and then initiating the movement-related cells. And this is an example of movement-related cells. These are different trials. Every line is a trial. These different marks here are spikes of that neuron. And so you see this is a neuron that modulates its firing rate in time when a movement is prepared and then initiated. And actually the initiation of the movement is here, this first bar, I believe, or actually the first bar is when the movement command was given, and the second bar is when the movement is initiated, I believe. And what you see here is that there are directions of movement. Each rasterogram is plots here in the direction at the location here that reflects the direction which this movement occurred. You see that these movements here elicit much more firing from this neuron than these directions. In fact, you see these directions here have a suppression of the firing rate over the spontaneous background activity of the neuron. So it's actually inhibited over its spontaneous firing rate. So if you systematically plot averages of firing rate across many trials as a function of the movement direction of the hand, then you find also a very broad, smooth tuning curve. In fact, it's very broad. This is the full circle, 360 degrees. And so this is like a cosine function. So it's a function that the broadest periodic function that you could actually think of that still is modulated with movement direction. And that gave us this idea of population activation, in that if a lot of cells are tuned this way, and this is quite common, about half of the cells that are movement related in motor cortex have this kind of tuning, then you find that for any given movement, at least a quarter, if not more, of the neurons in motor cortex are actually quite active because of this width of the movement, the tuning curve. So this is how the coding, the input-output relationship is envisaged, or is characterized in this classical form of physiology. Now, when you do this from many neurons, then you can look at how many neurons together represent the state of the motor system, or the state of the perceptually the perceived environment. And that really adds a quality to that relationship between inside and outside. So it's not just that you do some averaging, there is actually potentially a lot more information about the world outside. So I'm just illustrating this here, that let's say you have 3 neurons ABC, and these neurons have different tuning curves, ABC, then you might naively think, well, every neuron just sort of encodes its preferred value, like its peak value, and then I have a pretty crude sampling, so it can only roughly estimate here if it's closer to A or closer to this or closer to that value. But that's actually not true, because if you have, for any stimulus, let's take here this stimulus, this particular value of the feature dimension, right? Now you have these 3 numbers, the firing rates of these 3 neurons, and these 3 numbers uniquely identify a value along this axis that is consistent with observing these 3 firing rates. So with 3 neurons, you're actually perfectly fine. You can actually be fine with 2 neurons. So it's certain patterns of activation in these neurons that uniquely encode a particular value along the feature dimension. So in the rate code picture, the issue is actually not so much how many neurons you plant here, but how precise is the firing rate. So the pattern of firing determines the value. That depends on how precise you can estimate that firing. So you need actually some redundancy in the system because of that, because firing is somewhat stochastic, and you need to get some reasonable estimate of the firing rate, just theoretically, right? So at the population level, a new quality comes in. It's not correct to think of populations as very crudely just sampling the underlying feature dimensions by the center of the receptive field or of the tuning curve. They actually can do much more. It's very common in the brain to have relatively broad tuning, and by that I mean that neurons will be active over a range of movement tasks and a range of stimuli that is larger than, let's say, the behavioral averaging. You can make a very precise saccadic eye movement here, for instance, or hand movement, or visually discriminate between different stimuli. And the differences between which you can discriminate and can show different behaviors are much smaller than the typical tuning curves of neurons. So in some sense, it's actually not right to think of one neuron voted for a tuning curve, because that would be too broad. And so you can ask yourself, is that a neural activity that is broader in a functional representation, is it actually contributing to behavior, or are you just taking the most activated neurons and just reading their response? And that would be then a limit, not really population coding. So population coding really would be based on using also firing rates that are somewhat low, not just here these top firing rates. And then you have this power of the population. And this is one of the very few examples. The original study that really established this by David Sparks' lab in saccadic eye movements, where you find similar tuning. So neurons are tuned to particular saccadic endpoints. So go over this area. This is a particular layer in the colliculus, superior colliculus. And these lines here indicate that neurons in particular locations along the surface of this system, when they are activated, then a particular saccadic endpoint is generated. You can actually electrically stimulate there and elicit saccades, and these saccades will go into a particular direction. And this is topographically organized. That is, neighboring cells will be generating saccades into neighboring directions. And that makes it possible to, in a very targeted way, manipulate these populations. So, for instance, if you've measured that around here, neurons encode or generate saccades in this direction, around here in that direction, and around here in this direction, then you can do things like activate, prevent in some way these neurons from firing. There's a way of doing that by, for instance, cooling these neurons through an electrode. You insert this little cooling liquid going through. When they cool, they can't fire. And when you do that, you can look if you're still able to get the correct the movement that would be encoded here by just activating this and that subpopulation, and you can. And more dramatically, if you cool a portion, so usually when you make this account to be, you will get this whole large population here to be active. But if you're not cooling a part of the activation, that it would be still somewhat active under normal movement, and then you will find that systematically your saccade is biased away from the saccade of B. So these neurons voted for the saccade to be, and you can see that if they're prevented from voting for it, then the saccade is a little bit off. It's closer to D than it is under regular conditions. So that really shows that all the activated neurons, or a very large part of the activated neurons, not just the most activated ones, contribute to the behavior. And there are similar studies in perceptual areas, for instance, MT, the area that encodes movement, visual movement, perceived movement, where you ask a monkey to estimate the direction of a visual display through some response. And then you can do very similar techniques and can bias the response of the monkey away from the actual response and stimulus by selectively deactivating some neurons that encode particular movement directions. This was done in a lab by someone called Newsom. I think I have cited that here. Yeah, this maybe this is not exactly the right census. So there is a general consensus that population coding is really a prevalent principle, certainly in the higher brain, in the cortex, so that you're both exploiting the higher representative power of populations, and you're also using these kind of non-local principles, so that entire populations contribute to the behavior. And so that DFT is really based on that idea. So DFT introduces these populations very naively modeled just by spanning these fields. And there were two sort of examples that we worked out in detail of how you can take neural data and then actually estimate the fields that the DFT models. And so the oldest is work that we did here in Bochum many years ago, before I went to Marseille, actually. This is Dirk Janke's doctoral work, who's still here at the ED now, a professor. Studying the primary visual cortex of the cat called A17. And it is really just a population representation of a retinal location. So in the primary visual cortex, you have these receptive fields that we talked about just now and previously, which makes that if any record from any given neuron, it will have a particular place on the retina where it will respond. And we established more precisely where that place was by roughly mapping that. And then we actually put a stimulus array that consists of little dots of light. Only one of those dots of light would be presented at any time. And doing that, we can sort of compute a receptive field profile. That is, we're plotting here how much the neuron fires in response to, this leftmost element, and then another element, and so on. That gives you this kind of response plane, and we smoothed that in some way. And then the trick was that we would also have some stimuli that we present to every neuron. Now, usually you only present stimuli to a neuron that it responds to, that is right on its receptive field. But we had some conditions in here where, when this is the fovea is here, this white line is sort of to orient us, and so we put certain stimuli, like this little bar, this little square here was lit up for a brief moment, and that stimulus was presented to all the neurons. And you will see in a moment, it's a whole line of different positions for that kind of light. And because we did that, we could now actually represent of how all these neurons together represent retinal location. And that was done, these neurons were recorded in different trials, and in fact, in this case, even across different experiments, meaning even different cats. This is from the cat, an S-sized cat, actually. And so the idea is that we're thinking of every neuron as voting for its receptive field. So the center of this is these spikes here. So that every neuron is put into the stimulus space at the location which is the receptive field center specifies. In a moment, you'll see we'll also use the profile. So these little needles here reflect where on the retina these cells are looking. In that sense, the receptive field is centered there. And the height of the spike is how much they're firing when this particular stimulus is given. And you see the neurons that whose receptive field is close to this, they fire a lot, and the neurons whose receptive field is far from that, they fire much less. That makes a lot of sense. So when we actually apply this whole field, we can make this into a continuous representation, and that would be an example then. But it's just superposing all the receptive field. This is a 3D plot. This is a contour plot you see that then the population, there's a population distribution over retinal space that sort of indicates directly how this population of neurons represents that visual stimulus. It's really precise only along this one line because we didn't sample the whole space. We only sampled, as you see from these spikes here, we only sampled neurons somewhat along this horizontal line because it's a lot of recording from it of a lot of different neurons. So in the experiment, we actually had different locations of the stimulus. And then we actually also, we called these elementary stimuli, that is, they were presented to all neurons. And in a moment, I'll show you stimuli that are composite from these elementary stimuli. So even if you do that, you can already observe, you can essentially observe how retinal location is represented by the population. So this is these are these 8 different positions of the square of light, and you see the population here in color code constructed by superposing the receptive fields, each weighted with the current firing. Then you see that this tracks the stimulus position reasonably well along the horizontal in particular. You can actually do more. You can take as this weighting factor not just the summed activation in the entire interval, but you can take different time slices of activity. And thus you can look at the temporal evolution of neural activation. And this indicated here, in 10 milliseconds time slices across the population, we can see how activation arises and then fades again. You actually see here the transient nature of the neural response to a stimulus like that. And you can ask questions like, does it come up narrow and broaden, or broad and narrows? And you see it comes up as it is broad from the start. And the more interesting aspect here is that we were able then to look at neural interaction. So really what the neural fields are about. So this was done by presenting now some composite stimuli, so stimuli that had 2 elements of light, and one was always the left one, and the second one was very close and then was further removed in different trials. And so the distance between the two stimuli was looked at, and we used this technique of the receptive fields and so on to represent how these composite stimuli were represented by the population. That's here the top line, and compared that to a hypothetical predicted representation of the composite stimulus if we just take the responses to the individual stimuli and superpose them, just add them, right? So this would be these to the individual stimuli, but we add the components for the 2 stimuli to predict what would be the population representation if it was just adding these inputs. So there was no interaction. It was just a linear superposition. This is, of course, not very good when they are very strongly overlapping, like here, because then the 2 populations will be strongly overlapping. So they reach firing rates that are way beyond what you see in the individual case. But here they're not overlapping. The individual distributions are very, very much like you see here. I mean, this one and that one are essentially an individual contribution. You see already with the naked eye that there is some kind of inhibitory interaction, that is this activation that is observed when both stimuli are presented is less than you get when the stimulus is presented alone. So there must be some inhibitory influence from that second stimulus. And this could be done more in more detail by a more refined technique. This is looking time resolved, so take the activation of different time slices, and we're just looking at the activation here at this leftmost location that was always included in the stimulus, and then for different distances of this other stimulus. And what I just pointed out was that when the second stimulus is far, then the real activation observed at the left, that's the solid line, is less than the activation that we predict at that location in the absence of this stimulus. And so here it's actually really just that, because there's no overlap, no contribution to this leftmost stimulus from the rightmost stimulus. So that is direct evidence for inhibition. And you see the inhibition arises here near the peak of this response and toward the end of that response. You see, again, see the time scale of this, starting with stimulus onset. And you see that signature also as you get a little closer, see it actually all the way through. The other interesting part of that is that there is this increase of activation somewhere here, for instance. So certainly here, but you could say, well, maybe here it's just due to more input, that this also provides input to that, and therefore activation arises early. But you see that even where the input from this stimulus is not very strong, you already have a little bit of that early increase in activation. So there will be evidence for early excitatory interaction, right? Which early, I mean, early in time, excitatory interaction. And that's for stimuli that are closer to each other. And if you remember back when we looked at this two-layer field, this is actually a signature of the interaction kernel in the field. You know, the one-layer field doesn't actually have excitatory and inhibitory separately, and therefore it cannot capture this fact that inhibition comes in later. But the two-layer field that I discussed in that lecture on selection, that actually accounts for that, the fact that inhibition comes in later than excitatory interaction. So we can directly in the cortex see the interaction kernel at work. Now, these experiments are not really showing a selection decision, right? Both peaks are present. So this is interaction is just a weak modulation. This is largely an input-driven response. It's not a self-sustained peak in any of the senses that we used. And there is actually recent work that hints at actual, like a detection instability, but psychophysical work, not neural work. And that's too complicated to explain here. Okay, and the model explains that, but I don't need to belabor that. The second story I want to briefly tell is the motor version of this. This is work that I then did with my colleague Alexa Riele and her student Annette Bastian in Marseille after I moved from Bochum to Marseille. A laboratory where there was motor neurophysiology and human motor behavior as a topic. And here we studied primary motor cortex. So the output station of motor cortex, like Jacopoulos. We used Jacopoulos' paradigm. We had the monkeys make center out movements. For us, it was a vertical plane where the monkeys pointed at buttons that lit up in different colors. We had only 6 directions for some experimental reasons. And in these experiments, there was always prior information, so some signal that told the monkey which of these 6 targets were coming up, either 1 or 2 or 3 were cued. And then at some later time, the response signal came where one of these targets changed color again, and the others were extinguished, and that told the monkey which movement to actually make. And the monkeys were trained to not move before that signal came, and, well, to go to the right direction. And you get this same kind of tuning, that is the firing rate is a function of movement direction. They're all neurons that are movement modulated, that is, your time modulation is aligned with the movement. In fact, you see here the one marker is here the liftoff of the button. This is when they land on the target. And you see that actually most activation in this particular neuron is actually before the movement is initiated. So motor cortex is in advance over the movement, as I hinted in the lecture about embodiment. So same kind of tuning. And now the population representation, that's analogous to what we just did, would consist of saying every neuron contributes its tuning curve, right? They have these cosine kind of tuning curves. It's just 6 values, right? I'm just showing an illustration if you fit a cosine through it. So these green things could be tuning curves, and the population representation will be constructed by multiplying each tuning curve with its current firing rate. This would be like they're taking the tuning curves as the base vectors, and you're building the distribution from base vectors. And then you get that red curve if you do that. So if this is if you're looking at this movement direction, the tuning curves close to that will be very strongly active. These ones and the tuning curves further back will be less active, and therefore the sum is then articulated like that. So this vector is this direction is the population vector, as Jobber's called that, and that will be the distribution of population activation. By taking the current firing rate in different time intervals, you can look at the time course of movement generation. So here, for instance, you see that there is already a little peak when the Q that we call pre-Q then enters, that already specifies the movement, and then as response signal comes up, then the movement actually, the peak really grows and the movement is initiated. In these conditions, monkeys were actually able to predict when the response signal will come, so the activation rises even before the response signal comes, because the monkeys had learned that it takes about a second to get that command, and they were already ramping up the peak. Here's an example of how the distribution reflects motor conditions. So here the distribution is broader initially when two movement directions are cued than if a single one is cued, as you can see. So it reflects really the information about the upcoming movement, and it can be modeled. And in the detection, or one of the earlier lectures, I don't remember exactly which one, I showed you these results from John Kalaska's lab that Paul Zizek obtained, where they looked actually at movement directions in opposite directions that were far enough to have bimodal pre-activation, and then they saw that how selection happens. That actually was not in primary motor cortex, but in premotor cortex. But the tuning is very similar. Yeah. So again, this can be extended. So the main point about this motor example, why I again belabored that, is that illustrates very clearly that when we now consider these distributions of population activation as estimates of a field, that it's actually not sampling the field, the dimensions by neurons, again, by their peak value, because every neuron actually contributes its whole tuning curve. So you could think of these fields as a bit more abstract. That's why I don't even like to talk about activation variables as neurons, because it's always a collective effect. At every location in the field, all kinds of neurons contribute that have overlapping tuning curves with that. In the motor system, these tuning curves are particularly broad, and that's why they the smearing out effect is not negligible. It's really a much more abstract picture. Forget about this abstract argument here. Good. So that's just a very rough neural embedding of these fields. So in the course, we're always thinking about these two ways the dimensions come about: input from the sensor surface or through the motor surface. In the section about embodiment, I talked about this direction, and I demonstrated how that direction can actually work on a demonstration of a real sensory motor system. That's as much as I want to put into this lecture.